From Microbe TV, this is TWiEVO. This Week in Evolution, episode 54, recorded on April 21st, 2020. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Hello again from LD Home Studios.
How long do you think you're going to be in that home studio of yours, Nels?
Yeah, upstairs closet here. Foreseeable future, I'd have to say. So, I guess some quick updates in my locality. In general, numbers are staying pretty good. Upticks here and there, but relatively stable. I would say a lot of hospitals and emergency departments, et cetera, are kind of like ghost towns still, which is a good thing.
Wow.
But, and so a lot of talk, a lot of chatter, as we're seeing in the news about, quote unquote, reopening or loosening kind of the stay at home situations that keeps me in the home studio. I'm a little skeptical that we're ready to open up shop, as some of the, I can see a lot of the impetus for that and the impatience, but I'm hoping, I think if we already look back in 2020, one of the key phrases that has really come bubbled to the surface is social distancing or physical distancing. I'm worried that if some of these things unfold, like we're seeing in some places, Georgia, for example, that second wave might become a phrase that starts to dominate our conversations if we're not careful.
Could be, yeah. I mean,
I don't know.
Why do you think you have so few infections? Has travel been restricted into Salt Lake?
Travel, well, so everything kind of shut down. I mean, it was a week or two, probably 2 weeks, I would say, after what we saw on the West Coast, obviously on the East Coast, in your region. Mm-hmm. But it's been, and actually at the state level, Utah is still one of the very few states that didn't issue a stay-at-home mandate. However, in Salt Lake County and in Salt Lake City, the mayors have put that into place. Wow. And people are taking that pretty seriously, have been. At the same time, we're seeing some of those small protests where folks are showing up at City Hall in this case in sort of big numbers. We're seeing actually the trailheads where people, especially this time of the year, as it's looking out the window here at all the flowers coming onto the trees, the leaves coming out. And so people, have a lot of pent up energy to get out on the trails. And so those could be locations where you could imagine that we would sort of let down on social distancing. But, so far, fingers crossed, so good in terms of the relative numbers. So I think I haven't looked at the website, the state website on numbers, but I think the last time I did in the last day or so, something like 3,000 cases, maybe 29 deaths so far in Utah recorded.
Interesting.
What's the latest from there?
Well, things seem to have peaked here, but it's hard, you have to go a few days out before you're sure. But I was looking at the curves for New York yesterday, and, there's a curve up, and now it's on the way down, but it's just a little bit, so it could easily go back up again. But we've been doing serious distancing here. Everything is closed, right? Yeah,
of course.
Today I saw an article from USC in LA County. They did a serological survey, and they're finding way more infections than they thought. They get 2.8 to 5.6% of the adult population has antibody. And so that's 28 to 55 times higher than the confirmed cases by PCR. How about that?
Yeah, I don't know. So is that the Santa Clara County study?
No, this is USC.
There's another one.
The Clara one has been discounted as flawed, right?
Yep. Where is this one? Is that one actually New York?
This is L.A.
Yep. Well, that one, if I'm remembering right, has so far only been released as a press release. So,
yeah, that's right.
And okay. So that one, I would, hopefully the raw data, or at the very least, the sort of manuscript level analysis will come online to look at that. Certainly I think those numbers wouldn't be surprising in locations where there's been tons of cases confirmed.
For sure.
But whether or not that applies to spots where things are a little more quiet, that's one of the things I'm hoping to see actually here in Utah, which I don't think we've seen yet, is that there is good testing capacity that's come online. But in some days, because the requirement to at least have some symptoms is there, a lot of possible tests that could happen every day aren't even happening. And so
you're talking about PCR tests, right?
I am. Yep. And so looking for the virus. So starting to get to better surveillance, that feels like a key piece of the puzzle, which is lagging in many circles. And then
that
the
LA study is antibody. Are you doing any antibody in that state? Do you know in Utah?
There is. So and it's sort of circles back to this kind of personal connection. So one of my colleagues, Clement Chow, who was a confirmed case here. I think it's still a small enough community. I think it was the 5th confirmed case, something in the state in the pandemic. And so there's a local company, I think it's called Med Sentinel, that has developed a serological test, and they sent 4 copies. It's almost looks like a pregnancy test, right? You've got instead of peeing on the stick, you put a drop of blood, and then you're looking for those bands, which indicate that your blood contains antibodies that react on the strip, and then that would be a positive. It sort of fills in a cross. And Clement was telling me that his, basically the second he put that drop of blood on the test, the band lit up. It was the strongest response they had seen in their testing so far. So he's actually now, I think, scheduled to do some serum donation, as that's being considered as a sort of stopgap intervention for folks who are declining quickly. So there is a little bit of testing here. That's still, again, early days. And then the big engines would be the ARUP laboratory, is a big diagnostic lab that's emerged out of the University of Utah. And then there's a second big diagnostic crew, BioFire. I talked to one of the former postdocs in my lab, Alicia McCune. She's a scientist there now. And they're working full time to get their SARS-CoV-2 tests online, which is a really high throughput. It's PCR based, so it's looking for the virus, but it's the turnaround is basically 30 seconds given the microfluidics that they use. And so they're hoping to launch that within a week or two here. Yeah. So
they make the biofilm arrays that they use for respiratory pathogens, enteric pathogens, all that sort of thing, right?
Exactly right. Yep. Yeah. That's the crew. Yep. So, and Vincent, I wanted to wish you a congrats on TWiEVO 600 that just happened. I guess that must already feel like ancient history, given how many.
Yeah. We're already at 700, man. Yeah. It was only 2 weeks ago. Thank you. And thank you for your kind card and the Luke Jerram sculpture, which I have.
Yeah. Has that arrived?
It's on its way. I got an email from DHL. It's going to be here any moment.
Oh, sweet.
Hope it's in one piece. I hope it's not it's not defective interfering particles.
That's right. That's right. I guess if it's smashed into small pieces, it's sort of positive tale about defeating the viruses by literally crushing them or something. But untitled future mutation.
That's
right?
so cool. That's very cool.
Yeah, I love that. Yeah.
Yeah. It's very good. It's very nice. Thank you very much, and thank you for being part of the team. It's amazing.
No, of course. Yeah, it's an incredible crew. I was happy to do a small part and pitch in with the roster. I think 16 strong.
That's amazing. I didn't even realize that. I never counted it, but 16 people. Yeah. Wow.
I didn't realize it either until that came together, and it was really fun to be a small part of that. So today, Vincent, we've got kind of a tasting menu. I have to say,
we do. I'm hungry, man. I'm hungry.
Hungry for some virus evolution. That's great because that's what's on the menu. So 4 preprints that will cover the first 3 SARS-CoV-2, and then the last one is we'll see again for our third attempt. We'll see if the third time is the charm. For a virus that's not a coronavirus, actually, and in fact infects critters that live, single cell critters that live in the water. It'll be a fun, maybe diversion from coronavirus as usual. But yeah, why don't we dive right in? So the first preprint, you lifted this one up. So it's entitled SARS coronavirus 2 replication in Vero E6 cells. We'll talk about what a Vero cell is. Replication, kinetics, rapid adaptation, and cytopathology.
Yeah, this was sent to me this morning by Amy.
Oh, great.
In the lab, who's always sending me good stuff.
Yeah, this is an interesting one. So Eric Snyder, I think, is maybe the lead. This is a group or a couple of groups in the Netherlands.
Yeah, that's right.
And they're using a SARS-CoV-2 isolate from Australia.
Interesting story there, right?
Yeah. So, what was going on there? I think you tweeted about that earlier today. What
I tweeted a line from the paper where he said, we tried to get early on an isolate of this virus, and we ran into difficulties. How did he put it? I have to find it here. Here. I guess I should just look for my tweet. Yeah, he said, we ran into administrative barriers to getting the virus, and he said, I don't understand this. So we went to our friends in Australia, and we got an isolate from them. It's kind of sad, isn't it?
Yeah, I guess this idea that somehow there's ownership to viruses.
Crazy. It's crazy. Yeah, here he goes.
Yeah.
When requesting virus isolates, February, and in spite of the rapidly emerging public health crisis, we were confronted not for the first time with administrative hurdles and discussions regarding the alleged ownership of virus isolates cultured from anonymous clinical samples, which is absurd because the sequence is out there, so nobody owns this. You could make your own virus if you want, right?
Yeah, of course. And getting a hold of viruses is already, there's a little bit of red tape in the way. So generally you have to go through a MTA, a material transfer agreement, getting the tech transfer, the lawyers in the university offices sort of agree to exchange materials. That can already be a hiccup. But there's no reason to add more roadblocks for exactly the reasons you're saying. So
they got it from their friends in Australia, and they said that lab has provided it to 17 other labs globally to help people to work with this, which is the way it should be, right?
Absolutely right. Yep. That's sort of the way science, that's sort of why science does work is, I would say across international borders, that notion of putting stuff out there, getting it in the hands of other people, having that conversation that accelerates the discovery process, of course.
I had the same issue with Zika when we tried to get some Zika viruses years ago. People were saying, oh, you can't, we have this paperwork, blah, blah, blah. So I tweeted about it, and then somebody offered to send me theirs. So you can use Twitter as a good medium.
That's right. It's a good way to break down barriers. I mean, this happens too. So in, evolutionary biology, especially in molecular evolution, where there's massive data sets, and then, people sort of try to control access. It takes a lot of resources often to generate these data sets, and and there are some good reasons that you would want to, sort of get some payoff from that. But the idea of investing all of these resources and then not sharing it so that other people could build from it has a similar kind of echo to what's going on in a lot of these cases. But so anyway, so these authors, they did actually finally get, as you said, that strain from SARS-CoV-2 strain from Australia. And then these are some interesting plaque picking experiments. And I guess right out of the gate, so they started putting these viruses into so the Vero cells, and this has showing up in a ton of studies, including more than one that we'll talk about on this TWiEVO episode. So Veros, this is a cell line. I was looking it up. We have some Veros hanging out in my lab. Currently just in resting comfortably in liquid nitrogen since we've shut down the engines. But Veros are originally, so these are kidney epithelial cells that I think back in the 1960s were isolated from African green monkey kidneys,
which we now call Vervet monkey.
Correct.
We can't. African green is no longer appropriate. Apparently, you have to call them Vervet monkey kidneys.
Yep. The Vervets. That's right. And old world monkeys. Vero, I don't think comes from Vervet. Actually, I think this is a different sort of history. I don't remember.
I don't know what Vero. That's a good question.
Yeah. And then the E6. So this was just a clone that was continuously propagated. I think it's currently available commercially from the ATCC, the American Type Culture Collection. They're probably flooded with cell line requests these days as people are.
I got the name. It's from a Japanese group. at Chiba University in 1962. The name comes from an abbreviation of Verda Rino, which means green kidney in Esperanto. Verda Rino.
Interesting. Vero.
Yeah. Isn't that funny?
Yeah. So the E6 in particular is a subset of the Vero cells that was kind of set aside or has continued to be really useful, especially for virologists, owing to the fact that it doesn't grow out of control. So a lot of these transformed cell lines grow like dandelions in culture. Like you start growing them on the in what you call monolayer, single cell layer on the bottom of a dish. And some of the Veros will just, like other continuous cell lines, will just start to overgrow. And so you get these sort of messy, more than monolayers, just sort of collections of cells, and they become a bit harder to work with, especially if you're trying to isolate things into the cell media, like viruses. And so because the E6s are said to be contact inhibited, as they start to crowd together on a dish, they actually slow down in their replication. And so as you propagate viruses in them, they're a little more well-behaved. And so that's why the E6s have been used. Veros in general have been a workhorse, I would say, for virology. In part because viruses, so one of the challenges, right, of making more viruses is you want to do that in a relatively safe cell culture system, away from having to infect animals or obviously humans in that category as well. And so the Veros somehow in their establishment into being a continuous cell line have lost a massive chunk of the genome on chromosome 12, which actually involves the type 1 interferon response. And so the viruses can do quite well because it's basically these cells have a hobbled innate immune system.
Yeah.
These details will, I think, actually become a little bit important as we consider some of the details of these studies that are showing up on the bioarchive. So this study using the Vero E6s in that Australian SARS-CoV-2 strain, they started getting plaques, which people have noted you can do in Vero cells for SARS-CoV-2. What they saw, and I think others have as well, is heterogeneous plaque size, small and large plaques. What is that? I've got a expert virologist with me today. What is that? How would you interpret those phenotypes on the plate?
Well, usually when you have a clinical isolate, which is not too far removed from people, you often see heterogeneous plaque sizes, which means they're genetically diverse viruses that maybe grow at different rates in the plaques and or some other. I mean, it doesn't have to be growth rates. It can be sensitivity to the agar differs among the different viruses. But because each plaque comes from an individual virus, it's showing you some heterogeneity. And now, if you picked one of the small ones and plaqued it again, it remained small, that would say there's some stable genetic property. If you picked the large one and it stayed large. But however, if you picked either one and they continued to be heterogeneous, then not clear what it would be.
Yeah, something's up. So here they took a small plaque, really simple experiment, in a large plaque, and then they just kept propagating it. Yeah. And then what they see, and even just in 3 passages, so these are 3 serial replications, meaning you grab that plaque so it started as a single virus, amplified or replicated to high numbers. You pick that, and then take that, put that onto another dish, infect, and that would be one cycle in the serial passage. And then you do that several times. And so when they picked the large plaques, they stayed large and homogeneously large. And then as they picked the small one, actually it started to shift. So instead of all small plaques, then you start to see heterogeneous, just like you're describing, and then more and more large plaques as you go. And so I think what that implies is that you're selecting for viruses. These are out replicating. Yeah. The large ones are out replicating the small. And then what you can do is look then, can you correlate this to genetic changes? Are there mutations that are changing in frequency in the population from the beginning of the experiment to after the 3 passages? And so they found they described some interesting differences. They didn't sequence the whole genome. And remember, as we've noted, the coronaviruses, these are really large for RNA viruses. in the neighborhood of 30 kilobases. I don't think they sequenced the whole genome, but they look specifically at the spike protein, where it's sort of the first place that you would kind of a high priority target in some ways, given that the spike, this is the protein on the surface of the virus that's recognizing the ACE2 receptor of the host cell to gain entry. And also because it's on the surface of the virus, this is, also sort of what the immune response, one of the first things it will see are the epitopes of the spike protein as well.
Yeah.
And so one of the interesting observations here was that they found some differences in the furin-like cleavage site. So I think on our first foray into coronavirus evolution, we talked quite a bit about about spike evolution and recognizing ACE2. We didn't have time to delve into this interesting thing about SARS-CoV-2 that differentiates it from a lot of really closely related coronaviruses that that are hosted by bats. And this is and it's not unknown to coronaviruses. You see this in others, but not its closest relatives. These furin-like cleavage sites. Can you remind us, Vincent, what that is?
So furin is the protease in cells that it's fairly ubiquitous, and the site here is recognized and it's cleaved by it. And for some viruses, so many viruses require cleavage of their spikes in order to get fusion. And for some viruses, like influenza viruses, furins are cleaving it, and that is often associated with higher pathogenicity. So the appearance of this furin cleavage site suggested that maybe, and this is not present in the closest bat SARS-like coronavirus that was isolated in 2013. It's not present there. So one of the ideas was that this maybe is what makes this virus able to replicate well in human cells.
Yeah, exactly. And so, so the host protease, the furin that recognizes the sequence, and it's just a little stretch of like 6 amino acids, mostly arginine. So have a basic chemistry to them, the polybasic site that then is recognized by that protease and then can facilitate, just like you're saying, the virus replication. So what's interesting about this is actually the mutations they see are altering or even deleting out that furin site. And so I think it's important to remember, like, exactly the point of, like, the host context really matters for how a virus might transmit or evolve to, almost in real time. And so here we're talking about the host now, instead of being a human patient, in this case, the virus isolated from one in Australia. It's now the Vervet cell line that is, E6, meaning that it's contact inhibited and also is missing most of its type 1 interferon response. And so we've taken things a little out of context here. And so you see these rapid adaptations. I think it's really, we do this approach in my own lab, and by taking it out of context and being able to quickly move through these experiments and then to correlate that with changes. But I think, sort of a massive grain of salt in terms of what does this mean in terms of natural replication dynamics or even natural evolution as it's moving through human populations. Yeah,
I think the fact that this is an really artificial system. I mean, you would like to say, well, I guess the furin site was not so important. And, a lot of people have suggested that, as I said, this was important for acquisition of human transmission. And it's also been suggested that the passage of some precursor in human cells might have led to the acquisition of the furin site and pathogenicity, and maybe this is a lab-produced virus. Well, I don't know how much you can take from these Vervet cell experiments, but you don't seem to need it, right?
Yeah, that's right.
And they cite another paper, which I looked at this morning. It's in Cell, and it is all about the structure and function of the spike. And they make pseudotyped VSVs. So VSV with the spike of SARS-CoV-2. And they find for the infectivity of those viruses, you don't need the furin site. You can take it out.
Yep.
Right. So, and I don't know, let's see what kind of cells they did that in. Let's see if they used Veros or Vervet. Let's see. Recognition architecture. Where's the furin stuff here? It doesn't say. Kinetic analysis. I'm looking for the figures. Functional receptor. Here we go. Am I getting close? I'll have to edit all this out. Damn. Maybe if I search for furin, that would help, right?
There you go.
We found the harbors a furin site and blah, blah, blah. What cells? I just want to know what cells. Sequence alignment. Yeah. Unexpected furin site. Introduction of a few. Okay. We designed a mutant lacking the 4 amino acid residue insertion of the Fuehrer cleavage site. Okay. So they put this in the pseudotype. And it didn't matter. Yeah, Vero cells, dude.
There you go.
Sorry it took so long. So
no,
it's all consistent in Veros. So the real question, of course, is whether this is still true in humans, but we don't know. At least in Vervet monkeys, you don't need the furin site to grow this virus, right?
Yeah, that's right. And I think from an evolutionary standpoint, it makes you think of trade-offs, right? So why is it so this isn't the first time a furin or these polybasic sites show up in coronaviruses. So there certainly are scenarios of transmission, and some of that evidence that you're describing of increasing the ability of fusion it's even beyond coronaviruses. This is a common theme that you see in other viruses.
Yeah.
But in the context of the Veros cell, this is a much simplified system where you're not going after the air, like you don't have to traverse the airway, right? With all of the physical barriers, the mucin-related stuff, all of the glycosylated sort of torturous maze that the virus needs to navigate. And so, sort of pulling that away, this might be exposing that trade-off where basically you see, and that's the first thing to go, is that you've sort of lowered the guard. And so the viruses, it does illustrate when you put real selective pressure on these viruses, how quickly they can change. But again, important to think about the context here. And in fact, looking at this study on bioRxiv, they go on now that they have established this in their lab, the Veros cell infection system and the nice plaquing, which we've talked about. You've spoken about eloquently how the plaque has driven virology, having that phenotype. This is the key ability to measure how viruses are replicating with a visual impact of each virus becoming, the invisible becoming visible. So here they take advantage of that to then do some tests of adding some drugs that have been proposed as anti-SARS-CoV-2. And in fact, the one that they sort of put forward as the most consequential, and they compare this to SARS-CoV. So the original SARS from the early 2000s, to SARS-CoV-2, the one that we're up against now. And they say of the 4 they try, so this is the chloroquine, which has made a massive impact in the news. Remdesivir, the Gilead drug that's also making ripples. They actually say in their hands, pegylated interferon alpha was the one that had the strongest impact. Again, we have to let's think about the context. So we just mentioned how in Veros cells, there's a massive genome deletion that impairs the interferon type 1 system, interferon alpha. And so putting back interferon alpha might just be perking up these cells a little bit, and it's all relative. So if you start with something that's really sort of, low in that part of the immune response and then perk that could probably explain that result relative to the other and sort of gives you pause that this is really sort of all of a sudden stockpile pegylated interferon for SARS-CoV-2.
Yeah. No, it is, the virus is sensitive to interferon. It's quite clear. That's why it grows so well in the Vervet cells. But, I think that's a good point about the sensitivity.
So then I think we'll keep moving. We're on our tasting menu here. So, it looks like you added another. I
added one, but let's do your patient one first. Then we'll come back to that. I think it makes sense because this patient derived is Vero also, right?
Yeah, exactly. And so actually, this is kind of fun. So if I could mix these two preprints together as a reviewer and bring back the plaque assay, I think that might actually potentially strengthen our second paper. So let me set this one up. So this one, another preprint. And by the way, these are just showing up like in the last 24 hours or so. So we're kind of on the bleeding edge here. This is kind of science by rough draft. And people are putting stuff out there for better and for worse, just really early stage, peeks at what they're doing in real time under, I would say, more usual scientific, sort of situations. these kind of studies would be considered really preliminary data that you would repeat, or you would get some feedback. You would do some other experiments. You try to you get some complementary support. Here, we're really just kind of looking at things that are rough. And so that is a caveat maybe across the board here. But so here's the next one. Title of this preprints: Patient-Derived Mutations Impact Pathogenicity of SARS-CoV-2. This is a collaboration out of Zhejiang University in China, Lanjun Li. And this one came across my radar from another author, Chao Jiang, who put this on Twitter in the last, I think, yesterday afternoon. And that's kind of when I saw it. And, again, so science by rough draft, but also there are some really good reasons to share this. We've talked about the bioarchive in the past. And one of the things that what this co-author stated was, we've got these results, and we'll talk about it a little bit. These are viruses derived from 11 different patients in China, and I think this is these are sort of early days. So these are isolates from January, maybe even to early February, and then taking these isolates and then infecting those same Vero E6 cells that we were just talking about at length, and then seeing pretty drastic differences in some cases with how well they think the virus is replicating, and then correlating that to mutational differences between the 11 strains. And so this gets at, there's a bigger evolutionary question here that tons of people are interested, that we've talked about a little bit, which is the virus evolving in real time to get more? Is it replicating better? Is it getting more pathogenic? Is it going to become sort of a super virus? And so, we've tried to put, I think, a little bit of balance into that conversation, that viruses change all the time. There are mutations that show up. As we try to interpret the phylogenetic trees that are showing up in real time, I think there's now, the last time I checked, there's now 10,000 strains that have been sequenced, that we have genomes of SARS-CoV-2. This is ridiculous, like real time, sort of advancing of genomic epidemiology here. But how do we interpret this? And so even it can look like we have these massive differences emerging because there's different clades or different groupings on trees. And yet when we sort of go beneath the surface, it's really just a handful of mutations. And that's the case here. And what these authors would like to know, what a lot of us would like to know, is this kind of what we'd expect, sort of virus evolution as usual, which is mutations happen, but have very little functional consequence? Are they neutral, or are they just sort of bottlenecking events? Sort of, you pull a marble out of the bag and that one marble will go on to infect the next person, but there's, potentially millions of marbles to pick from. And so it's this random process. It's not that natural selection is saying this is the right marble to go forward because it will outcompete the other marbles. And so what the reason this one I think is kind of catching a lot of people's interest is they report what they're calling a 200-fold replication difference based on some PCR assays. We'll dig into that a little bit. And then some I would say some rough observations about CPE, or cytopathic effect. When you look at that dish of the Veros cells, you see that they look sick. They're not doing well. They're starting to ball up, round up. They're not holding on to that dish in that clean monolayer. They're feeling the effects of the virus replicating through. So let's see. Let me pull up my here we go. The figure here. So first, to just actually consider a little bit of the phylogenetic distribution. So in one of the so they've taken these 11 strains, sequenced the genomes, and I would say it's kind of what you'd expect. So maybe 6 to 10 mutations, and some of them are a little bit intriguing. So they're non-synonymous changes, meaning you change the DNA, or the RNA, I should say, the nucleic acid, and the resulting mutation leads to a difference in the amino acid. There's some mutations in the spike protein. So I think one that's kind of catching people's eye is a substitution D614 to G in the spike protein. This has been associated. Then you can also, obviously, and we've talked about this with the next strain data and related stuff, you can correlate where these strains are coming from the location that they are collected. And that mutation, for example, is mostly isolated in European populations. I think there's been some pretty credible suggestions recently that the stuff that's happening in New York, out on the East Coast, a lot of those introductions were sort of a two-step process. So the virus originating back sometime late 2019 in and around Wuhan, China, then moving up to Europe, and then with a lot of the cases from Europe then to New York, which would probably reflect, obviously a massive travel corridor, versus sort of a one-step move, which is from China to the West Coast, in Seattle in particular, where a lot of the genomics is happening. And so there's sort of, in some ways, a lot of these 10,000 genomes. There is certainly in the early days, preponderance of them coming out from there. So anyway, I think we'll see more about this. But what the authors here have done is shown how even if they take their 11 patients, all from China, and I don't think it's Wuhan, I think it's a different part of Eastern China, but with some travel again before things really shut down. And you can already see that even in this sort of small collection that these end up associating on very different parts of the tree. I think what you'd really be interested in seeing, given some rumblings on Twitter and other places, are more of the strains isolated out of Europe, maybe being put through the paces of how they're replicating. But what the authors do is, so they know that they have kind of the sampling of several mutations, and as I said, some interesting hints. So, for example, I think they mention two of the strains where you have a different point mutation. So the nucleic acid is different, but it leads to the same substitution of an amino acid in the spike protein. And so that's sort of, two different routes basically to the same outcome, two mutational routes. Given the degeneracy of the genetic code, you can have different codons that actually encode for the same amino acid. And so when you start to see things happening more than once, right, this is sort of that bedrock idea of evolutionary biology, that convergent evolution or the independent emergence of things are generally sort of, you kind of perks up your radar that something functionally interesting might be going on. So anyway, they move to the Veros cells, and what they say is that as they infect the cells, and they do a time course from 0 hours, I think, out to 24 hours or maybe even 48 hours. And then as they collect samples as they go from these infections, these time courses, they do PCR. So they're trying to see, can they amplify different parts of the virus genome at different times? And I think I was listening to part of TWiEVO. It's hard to keep up. I can't keep up, but I caught a few snippets there. And I think Rich Condit was describing or comparing these PCR assays to a golf game. If your score is lower, that means the virus is doing better. The virus is winning. And that's because you're detecting the virus particles earlier in that process of amplifying them through PCR. And so what they show very clearly is that the longer they go in their experiment, the scores get lower, like a golfer doing really well. But the thing that they really wanted to point out is that among their 11 strains, they actually see up to a 200 fold difference that they're inferring from those scores being lower for different strains. And then they say this also sort of connects to the cytopathic effect. That is, if you see more virus as judged by the PCR, the low scores in the PCR, that the cells they look more sick, that they're being impacted more by this. So I would say a few issues here. So, credit to the authors here. I actually have the tweet that kind of caught my attention. It says, just submitted this preprint, outstanding team, collaboration. Results are preliminary, but it concerns evolution and changes in virus copy load, cytopathic effects. Feel free to tear it apart. And so it's actually that last sentence, which I think really gets at it's offered, I think, in a good spirit here, which is, hey, this is a rough draft. Help us figure out what's wrong with this. And so immediately, actually, there's an interesting conversation that's emerging. There's a postdoc, Louise Moncla, I haven't met her, but she's in Trevor Bedford's lab at the Fred Hutch in Seattle. And she notes, hey, something's confusing here. I was looking through. So in particular, this mutation at position 614, this G mutation. And I think they've been looking at that. They're pretty interested in this coming out of this lab in Seattle. It turns out the strain strain virus number 1 from that set of 11 patients doesn't have that mutation, although it's sort of unclear based on the description in the preprint. And so obviously this is like very rough draft science. I would take it with a pretty massive grain of salt that again, some of the issues we were describing for the first paper were in Vero E6 cells, were in Vervet monkey cells that are already have a hobbled immune system, and to interpret what these mutations in that context might mean for just sort of a collection of patients that you pull out that where this virus is obviously circulating in humans, I wouldn't overinterpret that at this stage.
I would say this is never going to see the light of day because there's no plaque assay in this paper.
Exact.
You cannot make conclusions based on PCR. Here's a scenario for what's going on.
Yep.
These mutations are causing the production of a lot of defective pieces of RNA, and that's 200-fold higher than the other ones. It means nothing until you do a plaque assay, and I don't know why they didn't, because they have the virus, obviously, so they do a BSL-3 lab. And if this got into any journal, I'd be really surprised because they need to do some infectivity assays before they make these conclusions. So the idea that, a change can cause a 200-fold increase in copy number, it could be totally irrelevant. It may have no increase in actual infectious virus particles. That's my point there.
Yeah, no, I agree with you. And that would be what I was mentioning. So if we could take that as a reviewer, could we take that first paper that showed the small plaques, the large plaques?
Yeah, that would be great.
Could we combine that with
the
second paper, which takes a little bit of the diversity, not just the one strain from Australia, but 11 strains from China, sort of puts it in a little bit of that context. That's exactly what you would like to see, I'd say, in sort of draft number 2, is bring those together to advance.
The other things they say is, even the cytopathic effects could be influenced by the number of defective genomes. To say viral load is up to 270-fold difference in the abstract is not correct. It's not viral load. It's
right.
Viral genomes. And they say these mutations are capable of provide direct evidence that the virus has acquired mutations capable of substantially changing its pathogenicity. This is a total misrepresentation of the results. All right. There's no pathogenicity in this paper. What there is cytopathogenicity. All right. Damaging cells, but there's pathogenicity refers to experiments in animals. So this, the press is going to pick this up and go, shit, the virus is mutating to be more pathogenic. I told you so. No, please.
That's right. Exactly. No, that's right. And this is sort of this is science by rough draft. I mean, there's so much energy right now, and understandably so, in this space, right? Labs are scrambling to try to get a little bit of a foothold of what's going on. And in one sense, I think this illustrates something really good about science, is that this is what you want. You want hundreds of groups going at this, seeing if they can get a handle on something that's real. And then what would usually happen is a lot of that process would be sort of out of the sight of the public eye, where you, like, for example, all of this stuff, whether it's a press release about serology in Los Angeles, whether it's, some more basic research like this, it would be, these discussions would be happening very naturally, and they do, but not kind of out in the open like this. And so we're kind of doing this in some ways, I think it's really useful, and that it kind of pulls back the curtain and illustrates how science happens. But there are some good reasons to keep the curtain in front at some points, and let the process play out so that these conversations exactly can happen. It might be 3 reviewers who would see this as this would be submitted to a journal, who would make some of these very same points. One of the reviewer 1 might say, "Wait a minute, this is in Vero E6 cells. How can we make these leaps to right?" Or your point. There's not even a plaque assay here. You can do that with these cells. Look at this other work that shows how you can do this. Maybe do that. Give me to actually support the notion that there is more cytopathicity here, that the virus, there is a higher virus load, and that it's actively replicating viruses. The plaque assay, the engine of virology, would help to make that case. Again, missing.
Yeah, for sure.
We're seeing all this kind of mad scramble, right? And yep.
I'm just concerned that this gets published in its present form, and then reviewers, because of the current situation, people are throwing critical thought out the window, and this gets published and it contaminates the literature. And I'm hoping
that
the reviewers are going to do a better job.
I agree. And so as the author says, feel free to tear it apart. And I think we're trying to do that in the spirit of how science usually would happen, in sort of less fraught times. But I agree with you, Vincent. I think this is a very big issue, as I'm starting to see. So I'm an editor associate editor with a couple of journals, and also, of course, like we all do, get invited to volunteer my time to review articles like these ones. And we're getting flooded with stuff that just doesn't usually isn't approaching the bar that I would say we would usually see. And so it is a time to be cautious about. So I'm actually very much in favor of sort of people working fast under the circumstances, getting things out there. But I think what we have to do in equal measure is exactly what we're talking about, is have the critical eye and not lose track of the standards that go into interpreting evidence through a critical lens.
Yeah.
Okay, so now, actually, let's circle back. So you added, this is an interesting one. I think this came across my radar a while ago. So this is another preprint. And by a while ago, I think I mean like last week or something like that.
No. Last week,
or last month, actually, in this case. I think it posted about a month ago. Excuse me. And the title of this one is "A Novel Bat Coronavirus Reveals Natural Insertions at the S1, S2 cleavage site of the spike protein in a possible recombinant origin of HCoV-19 or SARS-CoV-2." So in this one, it's another group, collaborative group. Eddie Holmes is involved. It's Weifeng Shi. So this is one of the, I would say, real experts in bat coronaviruses. And if I'm remembering right, this, people obviously, and this has an echo from SARS-1 and from MERS, is to try to get a handle on where did this virus emerge from. And, we've tried our hand a little bit at this in my own lab, is to try to to do the comparisons, the sequence comparisons or the genome comparisons between closely related viruses. And so in SARS, what happened as more sampling was done, it was possible to say, okay, it looks like you could almost put together circumstantial evidence for how the virus emerged, that it came out of bats. There was an intermediate host, the civet cats, and then it moved into humans. For MERS, again, starting in bats, moving through camels, and then coming into humans. Here, we've heard some talk, I'd say it's died down a little bit, but about pangolins as a potential intermediate host. And so this virus sequence doesn't put that to rest or add more evidence one way or the other, but does illustrate some more of the activity at that the S1, the S2 cleavage site. This is right by, really close to that polybasic site, and in fact starts to help maybe bring in at least a little bit of evidence about what's going on in terms of recombination. And so I think if I'm remembering right, in our first TWiEVO, virus evolution by land and by sea and by coronavirus, we talked a lot about that recombination event. So the amino acids in the spike protein S1, which actually then recognize the ACE2. And so there's actually been, I would say, 2 sets of papers that have emerged, kind of like these on the bioRxiv, some of which are trying to say, there's a recombination event. And it's kind of intuitive that you would think that recombination happened in S1 and that somehow facilitated the emergence from bats, perhaps still a unknown intermediary, or it could just be from bats, of SARS-CoV-2 into humans. If you look at it, though, and put that into the context of the other viruses, in fact, the ancestral version is the version that you see in SARS-CoV-2, meaning that you see that in a lot of the other viruses that have come from bats. And so the one that was known, I think it's called rat G13 or something like that, the one that was thought to be the closest relative. In fact, we and a few others what we think we're seeing is that it's the other way around. It's that guy that had a recombination event in S1 and went away from what most of the bat viruses look like. But because it was the closest one known, that would be sort of it would sort of draw you down sort of a different route that way. So this one, if I'm remembering right, this is another virus that sort of helps to helps to make that case. If it's the virus I'm thinking about, this is one that was on the. So a lot of people are depositing these virus sequences. Not everyone, but there's 10,000 now that are on this GIS-AID GISAID resource that was up until SARS-CoV-2 is mostly seasonal influenza sequences as they were circulating around the world for kind of real-time surveillance like this. I believe this virus sequence was actually removed at a later date. And this starts to highlight how, some of the politics, we were talking about this a little bit at the, before the show, this idea, half-baked idea that this somehow isn't a natural event. This has escaped a lab, or that it was, so I think the first sort of red herring was that, these are, rogue mad scientists that have released a virus. Then it was, they didn't, or have engineered a virus and released it as almost like a bioweapon. That's kind of died down. We're kind of a second wave of, I think, really irresponsible reporting and sort of inferences without a shred of evidence that this was released from a lab, maybe by accident. It's just now you've gone from these mad scientists who are trying to ruin the world to just incompetent scientists who can't keep their viruses in the lab. We're just seesawing, I think, with these really sort of crazy ideas with very little, or in this case, no evidence. And so that in some of these cases, there's a response, which is scientifically, instead of sharing data, if it's being misinterpreted or used for political reasons, then all of a sudden, very quickly, things start to lock down, and information that was being shared freely, which is we're already, I think, discussing how important that is in terms of getting a hold of these viruses, starts to wither on the vine.
Yeah, this one is interesting because this is a different group from the Institute of Virology in Wuhan. So they had originally isolated RATG13 in 2013 from Yunnan in a cave, and some people have said, they're working with that, and that somehow became SARS-CoV-2 and it got out, which is ridiculous because it's far enough away from SARS-CoV-2 that it's not the case. But they're still blaming that lab. However, this is a different lab, and this sampling was done in 2019 at a different site, and they pick up a virus that part of it is even more identical to SARS-CoV-2 than RATG13. And so it deflects the attention away from the Institute of Virology in Wuhan, I think, because you can't just say it's one lab doing this. It's crazy. This one has 97% identity in the 1AB gene, which is the first part of the genome that encodes the RNA polymerase and accessory proteins. So it's the closest in that part of the genome, whereas in the receptor binding domain, they say this probably would not even bind ACE2. So this is obviously recombinant, which is just emphasizing the fact that there are pieces of SARS-CoV-2 in bats all over the place. So the RBD in RATG13 is pretty similar. The 1AB here is pretty similar. There are pieces that probably recombined at some point and gave rise to SARS-CoV-2. And that's what we're looking at here.
Yeah, that's right. The other thing that for me kind of doesn't hold up with the whole lab escape idea is that if that's true, there'd probably be already reported a virus that's basically identical to the one that's circulating now. If this is just incompetent scientists who have cataloged all of these viruses that they've been working with for years, in a lot of cases, stretching back to 2010. And yet there's nothing like this on the record. There are parts like we're seeing here, and then these recombination events. But at least so far, all of the evidence is so easily explained by a natural release. And so, as soon as there's some evidence to counter that, if it ever comes forward, then have the conversation. But until then, this wild speculation that gets politicized, I think, is really pretty dangerous.
Well, this is, as Alan Dove said on TWiEVO Friday, it's an accusation. It's not a hypothesis because there's no data to support it. We have data suggesting that this is a bat virus, that's better than them saying, they had it in the lab and they released it. Well, to give us the evidence for that. All you're doing is saying they're sloppy. That doesn't mean a damn thing. Yeah. I was talking with Eddie Holmes over the weekend about this, and he actually had sent me And he said, nobody's paying attention to this paper yet. Here is a part of which is even more identical from a different lab and a different sampling site. So it is pretty crazy that people are thinking, and Nels, what I think in the end, they really don't understand the genomics and the phylogeny and the implications, and they just want to accuse.
Yeah, I agree 100% on that.
And what else Eddie Holmes said? He said, same thing happened for HIV-1, and the only thing that got rid of the speculation, people said it was a lab-made virus, was wildlife sampling that showed here's where it came from. And he said, that's what we need here that we don't have yet.
Yeah, no, I agree. And hopefully more forthcoming data that starts to fill in some of the gaps. But I think what it also illustrates too is something that kind of can get lost in the shuffle, again, from an evolutionary standpoint, which is how important recombination is to the evolution of coronaviruses, right? So we see these strains where you're kind of piecing together like, okay, here it's 97+% identical, but at this other part, it's only 80% or identical, and then sort of flip-flopping as we infer recombination events. A lot of the genome sequencing that's being done now, so those 10,000 genomes, which are a great resource, are really focused by and large on the point mutations. And that's happening, as we've described, and as we'll get into a little bit more here. However, the kind of under the radar are all of the recombination events that are happening. And so those, because of the way people are adding or taking these patient samples and then doing the genome analysis and then adding it to the collection, very good at finding the point mutations. Mm-hmm. However, the sequencing technology that's being used, and even in a lot of cases, the computer algorithms to just quickly try to assemble the viruses and put it together, might throw out or miss altogether recombination events. And so, and if you just sort of browse through some of the virus sequences that are available, you see all these regions where it's just listed as NNN, which is unknown nucleotide, one of the four, ATC or G or AUGC for RNA. And those things that are just sort of hiding in plain sight could be some of the most interesting evolutionary events or the interesting cases of variation arising in real time that are sort of pushed to the margins because it's just, what we can see are the point mutations, but not the recombination events. Okay, so then as our tasting menu continues, I wanted to put this one up actually. So you just mentioned Eddie Holmes. This is he and a couple others, Andrew Rambo, Oliver Pybus, and a few others in the UK and Australia. Not a lot of actual science here, but I think it illustrates one of the challenges here, which is with 10,000 plus genomes and trying to sort of catalog this variation. How do we even talk about it? So this preprint, a dynamic nomenclature proposal for SARS-CoV-2 to assist genomic epidemiology. And this sort of builds off of some of the conversations we had last episode in terms of looking at next strain. How do you interpret these trees? And so, if we were looking at sort of traditional phylogenetic trees or evolutionary trees at the species level, in some ways those are easy to talk to. If it's if there's a clade of or a group of cats in one area and a group of dogs in another area, those definitions are pretty clean. Here, because these viruses are still nearly identical, it's just a handful of point mutations that are being cataloged and sort of put onto these trees. Ways of trying to just describe that in more neutral terms, in a sense. And so they propose A and B as the major lineages. So, and remember, the A and B lineages have a common ancestor that we can trace back a few months ago. I don't know the exact latest, November-ish of 2019, in and around Wuhan. And then a lot of the sampling that was done then on the West Coast, Seattle in particular, given just the resources that were there, they're proposing calling the A lineage. And then the B lineage are these ones that went from China to Europe. And then we're seeing all kinds of sampling coming up on the East Coast of the US. And then they further break it down. So it's the A lineage, and then they do three sort of slicing. So you could be 0.1, 0.1, 0.1, and then, sort of derivations as you get even closer. But I think so it's sort of a technical note or a way of talking about this. I don't know if it will be actually widely adopted or not, but the reason I wanted to put it out there is just as a way of how do we even talk about this? Because it's really, so the trees look kind of intuitively familiar as we might think about species trees. But there's so little genetic variation actually here. And so I like this idea of sort of getting away from those, traditional ways maybe of talking about it that might imply that there are some really massive differences to something a little more subtle or just that reflects that there's actually very little change in these trees, something a little less provocative than sort of a species level difference.
Yeah, I don't know who something has to just gain traction like this, right? Because there's no official body to say, yeah, this is good. But it certainly is helpful to talk about a lineage like this and helps you narrow it down to where it is and so forth. And
that's right. Yep. And I think this is going to be, so I, kind of hinting at some of these rough drafts of science that we're considering today. I mean, there are going to be more and more studies, there's no question about it, that are going to try to link functional differences in how the virus either transmits or replicates, given the genetic differences that are being cataloged. And so, I think a couple of cautionary notes that are sort of coming out of our conversation are, one, what is the context of the infection? It's really easy to infect a Vero E6 cell. That's why they're used to propagate the virus. That's probably the best use for it. However, to try to interpret the virology in that cell line can fall pretty flat versus, what's happening in real time as the virus moves through the populations of our own species. One of the things I'd be interested is as we're hearing some of the clinical reports that you're putting out there on TWiEVO with Daniel Griffith and his parking lot reports from the hospital and some of the symptoms that are coming out. I don't know how many of those sort of high, frequency, lots of cases, how much genomics is being done. Are people sequencing those viruses from the patients as they're taking notes on the types of treatments that it's going through?
Probably not.
Yeah, probably not. But that, to me, that would be a place that I, as if we're going to try to link some of the genetics, as we're seeing in some of these papers in Vero cells, maybe, at least trying to correlate that with some of the clinical features that are coming out to see if there are any common patterns. That to me would make a little more sense. Yeah. And then putting it in the context of the different lineages, the A lineage, the B lineage, that we're seeing, and potentially broad strokes from differences in what's happening on the West Coast and happening on the East Coast of the US right now.
One thing I would really like to see, instead of using Vero cells, is to use airway lung epithelial cells. It's hard to do, I know, but that's really that's the relevant biology to this infection, right? And so
absolutely,
maybe if you did that, you'd find no difference in any of these isolates, end of story, and use virus infectivity assays to measure. That cell culture technology has become very sophisticated. And so
that's right.
Except for growing and titrating viruses, Vero cells don't really make much sense in studying the biology of these viruses.
I agree with you. Yep. That's right.
I think you got a good thread there. I would call this episode Rough Drafts of SARS-CoV-2 Science.
Yeah, let's do that.
And now,
can't just keep calling it part 3, part 4.
Can't go on forever. I would say, Nels, let's go to, we have 3 email here, which will take us 20 minutes. I would say let's do that and wrap it up. What do you think?
Yeah, good point.
Then you can go skiing.
That's right. There's still some snow. All the resorts are closed with the stay safe, stay at home, but you can do backcountry. So if you like very well distanced, there's still a little bit of snow left. Absolute. Well, let's do that, Vincent. So let's move, and we'll one day or another, we'll get back to our
it'll be there.
regularly scheduled programming. Okay, let's open the email bag. So Nathan writes, Vincent and Nels, it's a beautiful day for staying at home in sunny Chapel Hill with few, if any, clouds in the sky. Temperature is 20 degrees centigrade or 68 degrees Fahrenheit scale. Thank you for an excellent podcast, and particularly TWiEVO episode 53. As a dedicated TWiEV listener, I've learned of the Next Strain website and enjoyed Enjoyed the video. Thank you for the clear explanation of the various views, the radial view, the clock view, etc. The mouse over to see the exact strain and its source. That actually, when you do that, you can see some of the point mutations that are being cataloged. We live in an amazing time for molecular biology. At my laptop at home, I can see a visualization of more than 3,000 SARS-CoV-2 genomes, now 10,000, where they each came from and exactly how many mutations are in each strain. Wow. Late in the show, you mentioned evolution in music. In 2015, I tweeted about a great paper using extracted music, big data, and algorithms to agnostically map the evolution of rock music from 1960 to 1990. The paper is open access in my TWiEVO listener pick. So not only an email here, Vincent, but we have a listener pick, a rare spotting on TWiEVO. This is great. The data shows 3 stylistic revolutions. In 1964, the expansion of soul and rock. In 1983, new wave, disco, and hard rock. And in 1991, rap and rock. Here is a link to this great paper building on our evolution in music thread that it's been kind of percolating in the background here. So this is a paper from the Royal Society, The Evolution of Popular Music in the USA, 1960 to 2010. Thanks for all you do. Be safe and keep up the great work. Nathan from Chapel Hill, North Carolina. Thanks, Nathan. Glad you're finding that Next Strain website to be useful and hopefully not overwhelming as we go into more thousands and thousands of strains. That's, coming up with ways of cataloging it or naming those lineages and to sort of interpret all those mutations or trying to see real patterns from just sort of the random process of mutation. It's interesting work and it's out there. It's accessible, and the more eyeballs on it, I think, the better. And so we'll definitely add this to the listener pick as well and continue to build our conversation about sort of the mixing together even evolution and music evolution in this case.
It's an amazing time for molecular biology, that's for sure, right?
Yeah, agree. Yeah, I mean, in some ways, this, the ongoing global pandemic, which certainly probably isn't unusual from ones that have echoed through, if we take that evolutionary view, over hundreds of years. But to be able to watch it unfold in real time with the technology that's sort of at our fingertips today, there's been nothing like that before.
Yep. All right, we have one from Victor. Thanks for your great podcast. I have a question regarding your estimates of coronavirus mutation rate. You sort of downgraded that on the podcast, rightfully so, considering how media react to those. But while nextstrain.org gives a 24.75 subs per year for corona, it also contains data sets for other viruses. So for flu, they have 3.53 E-3 per site per year, which matches well with the literature, I think. However, when comparing apples to apples, it is 8 E-4 for corona, or roughly 4 times less the mutation rate of influenza virus. While this is definitely smaller, still pretty much comparable, don't you think? Thanks again for your great podcast, an essential part of my shelter-in-place routine these days. Victor, Moscow, Russia, a student of Vincent's online courses since 2014. What do you think, Nels? Is that pretty comparable?
Yeah. Well, I think it is a lower mutation rate, and we've talked about this a little bit. So one of the things that coronaviruses do, which is different than influenza, different than most RNA viruses, is they have some proofreading going on here. We've talked a little bit about how this might reflect the fact that these are kind of the super tankers of RNA viruses. You've got 30 kbs worth of RNA to handle, kind of a set of extended set of genes that are necessary to navigate the immune systems to allow the virus to sort of address the host cell and replicate. And so there's some level of purifying selection or gene conservation that comes with that proofreading that would then lower the point mutation rate per site per year. And so I think that's consistent with they aren't that much different. I agree. But the trend of it being lower in coronaviruses is certainly consistent with the biology here as well. I think, we've talked about it a little bit too, is to keep in mind the complexity of mutations. So the point mutation, again, we really focus on that, and for good reasons. It's a lot easier to measure, to catalog, to measure, to consider. A lot of the tools in molecular evolution are just, it's easier to use the point mutation to gain really sort of ideas about the evolutionary process. And yet, a lot of viruses, and as we've discussed as well, have multiple routes to gaining variation. So obviously in influenza, reassortment, right? The segmented genomes, the reason that we have the H and the N with the different numbers as these things reassort as sort of an engine for influenza evolution. All the recombination going on in coronaviruses, including even potentially horizontal gene transfer from very diverse sources. This is something that I think is also sort of important to remember. So even as the mutation rates look mismatched, as you sort of bring in all of the different mechanisms, these viruses are competing in the same space, right? They're up against the same immune systems. And so there is probably a pretty comparable level playing field, but it's sort of dialed in different ways with different flavors of mutations, depending on the type of virus that you're up against.
Yep.
Yeah, thanks, Victor. Really good question there. So our next one is for emails from RT. What a pleasant conversation about the science of COVID-2 and beyond. The measured and knowledgeable voice of Dr. Aldi through the welcome nudges of Vincent provides a welcome window into our current context and future challenge opportunities. Do you think, did I secretly write that, Vincent? Does it sound?
No, you're pretty modest, Nels.
Thank you for this conversation that welcomes laypersons into it. You both are so well-spoken. Impressive. Thank you, Nels, for your depth of knowledge, experience, and research in this important science, RT. Well, I'll take an email like that any day, Vincent, from up here at LD Home Studios in the closet here. No, that's very generous of you to say. And I have to say, and as we mentioned a little bit at the top, so just thinking about those 600 plus now episodes of TWiEVO and all of the people, all of the podcasters that have come together that Vincent has organized and recruited and partnered with over now more than a decade. I mean, that what you've built, Vincent, is a pretty incredible group of folks who are well positioned to have conversations like this. And so for me, it's a fun honor just to be in the conversation. Yeah,
we are positioned to be the ones having these conversations, right? It's just amazing, and really no one else does this to this extent, but I think it needs to be done. So I'm really happy to be able to get all these people together because I sure couldn't do it myself. That's for sure.
Absolutely. And as just a small kind of part of it, it's I'm really glad that folks are finding this to be useful, and as we're going, and stay tuned. So I think there certainly will be a lot more SARS-CoV-2, certainly in the near future, from sort of all channels, but there is more science out there, and that's still on our radar too. And
yeah, for sure.
And that will start to come back into the conversations as well. A lot of reasons that viruses are important, not just from current events from an evolutionary standpoint, but all kinds of research that complements that, supports it, builds it in different ways. And I think colliding these conversations in new ways and out beyond sort of the traditional, seminar kind of within a university, but to try to share these conversations more widely and to have it be two-way conversations. I mean, we were kind of bemoaning earlier on just how some of the stuff gets politicized or on very thin or no evidence stories take off in our news for political reasons. I think the more we can share in these conversations in bigger and broader circles, the better off we all are for stepping forward in a more evidence-based and sort of realistic way.
Good way to wrap up this show, Nels.
All right, we'll leave it there for now. Vincent, I do have one science pick.
Okay.
That I can't resist. So this is, if you're looking or hungry for some science beyond SARS-CoV-2, right now there's a great seminar series that has a YouTube related seminar series, the Evo Eco seminars. ongoing. So there've been a couple of seminars already. We'll put up the link. You can subscribe. So these are hour-long seminars by thought leaders, leading scientists in both evolutionary and evolutionary ecological biology. I think the first two, one was a friend of the show and a friend of ours, Harmeet Malik, just gave a seminar. And there's these, you can watch them live streaming, but then they stay around for about a week or so you can still catch up. And Molly Przewierski from Columbia also did I think she led off the series maybe a week or two ago. I think that's still available. We talked about some of Molly's work very early in TWiEVO history, maybe episode 2 or 3. And so check this out. There's a whole roster, I think, of about a dozen different great evolutionary biologists and ecologists who are signed up for this. It's organized by grad students. I'm blanking on the details right now, but you can get that information as well. And there, as the seminars are going on, you can actually, on a Slack channel, raise questions that then in a question and answer session at the end. So a really great opportunity to sort of brush up in various different areas in evolutionary and ecology topics, modern topics that are being shared more widely.
Well, now since you have a pick, I'll add one.
Fantastic. What do you got?
So this comes by way of Mike Buchmeyer, who sent it into TWiV, so I'm stealing it.
Fair enough.
It's a free book on the Apple Bookstore, Coronavirus, a Book for Children. Looks great.
Yeah, I agree. Great idea.
Explanatory and nice illustrations, and it's free. What more could you ask for? I'll put a link to that in the show notes.
Thanks. That's a great one.
All right. That's TWiEVO. You can find it on any podcast player. Please subscribe so you get every episode, and we know how many people are listening. Makes us feel like we're reaching people, right, Nels?
That's right. No, it's really great to get this feedback.
And if you want to see the letters and the links that we talk about, microbe.tv/TWiEVO. Questions and comments, TWiEVO at microbe.tv. And if you like what we do, consider supporting us, microbe.tv/contribute. Nels Eldey is at cellvolution.org. Over on Twitter, L Earlybird. Thanks, Nels.
Hey, thank you, Vincent. Good to be podcasting together again.
I'm Vincent Racaniello. You can find me at virology.blog. Music on TWiEVO is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back soon. Until then, stay curious.